Autophagy 2016 Nobel Prize and the way stem cells self-eat to rejuvenate autophagy, also known as the cell's self-eating process, is a miraculous biological mechanism that has attracted the attention of the global scientific community, especially after Professor Yoshinori Ohsumi was awarded the 2016 Nobel Prize in Physiology or Medicine for groundbreaking discoveries in this field. To better understand autophagy, we need to go back to basic biology. Every cell in the body has the ability to clean up and recycle damaged parts, helping to maintain life and the body's youthfulness. Autophagy, meaning self-eating in Greek, with auto meaning self and phagy meaning eating. This process occurs when the cell recognizes parts that are damaged or no longer functioning effectively, such as damaged mitochondria, faulty proteins, or components altered by oxidative stress. The cell encloses these objects with a special membrane called an autophagosome, then transports them to the lysosome to digest and recycle them into small molecules, continuing to serve life activities. In-depth analysis of the molecular mechanism of autophagy. To understand more deeply, we need to move beyond the trash bag image and delve into the activity at the molecular level. Autophagy is a molecular dance that is extremely precisely coordinated, with two main switches controlling the entire process: mTOR and AMPK. mTOR, mammalian target of rapamycin. Think of mTOR as the cell's satiety sensor. When the cell is full of nutrients, especially amino acids and glucose, mTOR is activated. This on state signals to the cell that conditions are good, focus on growth and division. Most importantly, mTOR acts like a brake, strongly inhibiting the initiation of autophagy. AMPK. AMP, activated protein kinase. Conversely, AMPK is the cell's hunger sensor. When the cell's energy is low, ATP levels decrease and AMPK levels increase, AMPK is activated. This signal means alarm, energy is running out. Alternative sources need to be found. When activated, AMPK promotes the process of autophagy and simultaneously inhibits the activity of mTOR. How does the dance begin? When you fast or do high-intensity exercise, cellular energy levels drop. AMPK is activated, and mTOR is turned off. This is the green light for autophagy. 1. Initiation. Signals from AMPK activate a protein complex called ULK1. This complex moves to a location in the cytoplasm and begins the call for bids process to build the autophagosome. 2. Nucleation. The ULK1 complex recruits another group of proteins, including Beclin-1 and VPS-34. This group produces a lipid signaling molecule on the cell membrane, marking the site to start building a crescent-shaped membrane structure called the phagophore, the precursor to the autophagosome. 3. Elongation and closure. Two ubiquitin-like protein systems, a type of protein that marks needs to be processed, are activated. The most prominent is the LC3 protein. Initially, LC3 is in the LC3-IE form floating in the cytoplasm. When autophagy is initiated, it is converted into the LC32 form and tightly attached to the membrane of the phagophore. The presence of LC32 helps this membrane elongate, envelop damaged cellular components, and finally close, forming a complete autophagic vesicle, autophagosome, with a double membrane. 4. Fusion and degradation. The autophagosome now moves within the cell and seeks out the lysosome, the recycling center. Thanks to proteins on their surfaces, these two structures fuse together, forming an autolysosome. The environment inside the lysosome is highly acidic and filled with hydrolytic enzymes. The inner membrane of the autophagosome and all the waste inside it are broken down into basic molecules, amino acids, fatty acids, sugars, ready for reuse. This process is not just random cleanup. It can selectively clear specific targets, such as damaged mitochondria, called mitophagy, or invading bacteria, xenophagy, demonstrating its remarkable sophistication. Evaluating the level of effectiveness. Why is there no exact percentage? This is an extremely important question, and the answer reveals the complexity of human biology. Providing a figure such as 16-hour fasting increases autophagy by X% is not scientifically feasible at the present time, and if it were, it would be an oversimplification. Here are the reasons. 1. Autophagy is a flow, not a water level. The effectiveness of autophagy is not measured by the number of autophagosums at a given time, but by the autophagic flux. That is, the rate of the entire process from the formation of autophagosums to their degradation in the lysosome. Illustrative example. Imagine a recycling plant. If you only count the number of trucks, autophagosums, lined up at the gate, you cannot tell whether the plant is operating efficiently or is congested. A large number of trucks could mean that waste is being collected very quickly, good flux, or it could mean that the processing inside is stalled, poor flux. Scientists face a similar problem when measuring biomarkers like LC32. 2. Measuring in humans is extremely complex, invasive. The most accurate measurement methods require tissue biopsy, sampling muscle, liver, etc., for analysis, which cannot be performed on a large scale and frequently. Indirect. Researchers often have to measure biomarkers in the blood, for example, in immune cells, but the level of autophagy in the blood may not accurately reflect what is happening in other organs, such as the brain or liver. Individual variation. The level of autophagy activation depends greatly on each individual. Adaptation level. A person accustomed to intermittent fasting or regular exercise may have a different autophagy response compared to a beginner. Genetics. Your genes affect the efficiency of the mTOR and AMPK signaling pathways. Age. Autophagy capacity declines with age. Metabolic status. The degree of insulin resistance, body fat amount, and baseline diet all have a significant impact. 3. What does the scientific evidence say? Instead of a percentage, science provides very convincing qualitative and comparative evidence. Fasting time. Most studies agree that autophagy begins to be significantly activated after about 16 to 18 hours of fasting and reaches its peak around 24 to 48 hours. After 72 hours, the body may start other protein conservation mechanisms. Statistically significant effect. Clinical trials, such as studies on obese individuals, have shown that the group practicing time-restricted eating, a form of intermittent fasting, had a statistically significant increase in autophagic flux compared to the control group after a few months. Significant here means the difference is large enough not to be due to chance, but it is not expressed as a percentage. Observable effects. Although the percentage of autophagy cannot be measured, we can measure its end results. Improved insulin sensitivity, reduced inflammatory markers, such as CRP, decreased oxidative stress, and improved cardiovascular health indicators. Conclusion on effectiveness. Instead of searching for a non-existent percentage, evaluate the effectiveness of autophagy through solid scientific evidence. Practicing intermittent fasting, e.g. 16-8, or regular exercise certainly has the potential to effectively activate autophagy in a way that is meaningful for health. This effectiveness is not a fixed number, but a dynamic biological response. It is strong enough to produce measurable positive changes in the aging process and disease prevention, but its exact level is a black box that science is still striving to decode. How does intermittent fasting activate autophagy? A scientific guide to hack aging. The cell's self-cleaning and recycling mechanism, autophagy. The cleaning process. When cellular components such as faulty proteins, aged organelles, or pathogens, bacteria, viruses, appear, the cell creates a double membrane structure called an autophagosome. This vesicle encloses and isolates these waste materials. Recycling process. Next, this autophagosome will merge with another organelle called the lysosome, which is filled with powerful enzymes. These enzymes will break down the internal components into basic units such as amino acids and fatty acids. Reuse. The raw materials obtained from this process will be released back into the cytoplasm to provide energy or be reused to build new molecules, helping the cell to repair and renew itself. This process is very important in adapting to stress conditions such as nutrient deficiency, pathogen elimination, and preventing the accumulation of toxic components that can lead to diseases like cancer, type 2 diabetes, and neurodegenerative diseases. Practical exercises in daily life. Understanding the mechanism of autophagy provides many valuable applications so that we can proactively improve our health. Below are methods that have been proven to activate and optimize this process. 1. Intermittent fasting. This is considered the most effective way to strongly activate the autophagy mechanism. When the body does not receive energy from food for a period of time, the cells switch to a survival mode and begin the self-cleaning process to seek energy and repair damage. Common methods. The 16-8 method. You fast for 16 hours and have an 8-hour eating window each day. For example, you can skip breakfast, have your first meal at 12 noon, and your last meal before 8 p.m. Eat, stop, eat. Completely fast for 24 hours, once or twice a week. Benefits. Helps cleanse cells, improve insulin sensitivity, reduce inflammation, and support weight control. 2. Regular exercise. Physical activity, especially high-intensity exercises, creates positive stress on muscle cells, stimulating them to initiate the autophagy process to adapt and become stronger. Effective types of training. High-intensity interval training. HI8 includes short bursts of maximum intensity exercise alternated with short rest periods. Endurance training. Exercises such as running, cycling, and swimming for extended periods also promote the autophagy mechanism. Benefits. Enhances muscle health, improves mitochondrial function, and strengthens the body's ability to resist stress. 3. Diet supporting autophagy. Some types of foods contain natural compounds that can help enhance this process. Foods to supplement. Coffee and green tea contain polyphenols that help activate autophagy. Turmeric. The active compound curcumin has anti-inflammatory properties and supports autophagy. Ginger has effects similar to turmeric. Extra virgin olive oil, rich in monounsaturated fats and antioxidants. Dark-colored berries, such as blueberries, containing strong antioxidants. Should be limited. Consuming too much sugar and protein can inhibit the autophagy mechanism. Therefore, a balanced diet with low refined carbohydrates is ideal. By applying these lifestyle changes, you can take advantage of the body's natural cleansing mechanism to maintain optimal health and slow down the aging process. Practicing intermittent fasting, for example, 16-8. We eat dinner at 5 o'clock p.m. and then have breakfast at 8 a.m. the next day, drinking water or diluted smoothies if feeling hungry, or exercising regularly definitely has the potential to effectively activate autophagy and is meaningful for health. I hope this video helps you balance your intake of nutritious foods and smoothies to achieve a beautiful, healthy figure. Support by liking and subscribing to the channel. Thank you.